If you ever have the misfortune of finding yourself on Twitter nowadays, you may have come across posts by this account, which features a scientist who looks like he was pulled straight out of Jetpack Joyride. His hallmark is wearing a lab coat, looking ominously at the camera, and writing vaguely threatening captions. Some of my personal favorites are ethics is holding back scientific innovation and progress, pursuing infinite potential is what makes our lives meaningful, and every pioneer or prophet must suffer. By all intents and purposes, this guy seems like an evil scientist. He's even hiring henchmen. When I first saw his posts, I thought they were coming from a parody account. But when I took a closer look at his name and the blue checkmark next to it, I realized I recognized it. Maybe he's been hacked, who can say? But as it turns out, I had learned about Dr. He Jiankui and his research in my college genetics class during our gene editing unit. His work displays the incredible capabilities of human gene editing with CRISPR-Cas9, but also exists at the forefront of the giant debate surrounding its utility. This video essay aims to address that conversation, but before we return to our resident evil scientist and his work, we need to talk about how CRISPR-Cas9 works to edit DNA. My name is Anna, I'm a biology and political science undergrad, and this is Human Gene Editing, the Cost of Perfection. So when I say CRISPR, I'm not talking about a minimalistic brand of potato chips. CRISPR is actually a revolutionary tool we learned to use to edit DNA. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The title just refers to a group of specific DNA fragments initially found in E. coli bacteria in 1987. At that time, little was known about why these fragments existed. It did prove to be an interesting question, though, because the same structure of genes associated with CRISPR were discovered in different strains of bacteria, and eventually similar structures were even discovered in H. mediterranei, which is in the archaea domain and completely isolated from bacteria. Although bacteria and archaea do come from a common ancestor, archaea diverged from that lineage anywhere from 3.1 to 4.1 billion years ago. So essentially, CRISPR has been evolutionarily conserved likely since around the time life on Earth began. Obviously, these DNA fragments must be extremely important to these microorganisms to stick around for that long. Fortunately, now we know why. As it turns out, CRISPR is essential to the immune systems of these different prokaryotes. These DNA fragments store a memory of any viruses that attack them by stealing their DNA and taking it for themselves. CRISPR uses this stolen DNA to create guide RNA so that when the same virus attacks again, the guide RNA can recognize the virus DNA, bind to it, and obliterate it. So, where does Cas9 fit into this? Well, Cas9 refers to a specific protein associated with the CRISPR system. Cas9 acts like scissors. With guide RNA as its guide, it cuts double-stranded DNA at a specific site. This allows new DNA to be added or removed from the chosen site. With an understanding of how this incredible system works, scientists were able to optimize it for human gene editing. It took time to assemble all the necessary pieces of the puzzle, including two RNA molecules, crRNA and tracer RNA, both involved in the guide RNA task of targeting DNA. But eventually, in 2012, the entire system was combined successfully for simple use by scientists Emmanuelle Charpentier and Jennifer Doudna. For their work, both women were awarded the Nobel Prize in Chemistry in 2020. This CRISPR-Cas9 system they optimized is incredibly efficient because it uses one combined RNA strand to locate the DNA that needs to be changed. All you need to do is change the RNA code for the desired DNA sequence. With this system, humans have been given an enormous gift, the ability to, for the first time ever, precisely edit human DNA. The question is, where do we go from here? Well, we've done a lot already. In a little over a decade since CRISPR-Cas9 has been optimized, we've used it to create a new generation of drugs to combat genetic diseases. In the US, we first treated a person for sickle cell anemia with a CRISPR-based drug in 2019. Now, the FDA has approved an entirely CRISPR based therapy for the disease as of 2023. This incredible drug is capable of fixing mutations in DNA through the use of CRISPR-Cas9, effectively treating the disease with single usage. Many more uses for CRISPR based drugs are in the works, including those for cystic fibrosis and other deadly genetic diseases. But CRISPR-Cas9 is capable of fixing so much more. Why stop at genetic disease? Why not make changes before birth? Why not cure genetically inherited deafness or Down syndrome? Sure, they might have vibrant communities with their own unique identities, but who cares? The technology exists, so why not use it? Now would be a good time to return to the good Dr. He Jiankui. Dr. Jiankui is a Chinese scientist who had a lot of questions about the full capabilities of CRISPR, as well as the easily accessible means to get some answers. He found couples with fathers with HIV and mothers without. He then genetically modified the human embryos from these couples with CRISPR-Cas9 in order to, allegedly, make them resistant to contracting HIV. In reality, the treatment only worked for one baby, a twin born from one mother in 2018. When Jiang Kui presented this research at a conference in 2018, he was met by an uproar of criticism by his fellow scientists. His own country wasn't a huge fan of the massive can of worms he unleashed either. The same year, he was sentenced to 3 years in jail and fined an equivalent of $429,000. Now, of course, he's out of jail and apparently wasting no time to get back to his research. One of the largest criticisms of his work with CRISPR-Cas9 was safety. He dealt directly with human embryos, called germline gene editing, which is significantly different from the somatic gene editing being explored by CRISPR drug therapies. In 2015, a separate group of scientists led by Junji Huang attempted to remove a mutation that causes beta thalassemia, a fatal blood disease, in 6 human embryos with CRISPR-Cas9. Instead, they only managed to remove the gene in one. In 2 other embryos, they removed the gene but also accidentally changed other genes. In the other 3 embryos, they were unsuccessful entirely. Germline gene editing is illegal in most countries because of these potential safety risks that arise from an increased risk of imprecision and the fact that any unintended effects can be passed down through generations. One of the greatest criticisms of He Jiankui's research is that because of a mistake in sequencing, there is no way to be sure if any off-target or unintended changes to the twins' DNA were made. There's also a question of consent. Obviously, it's impossible to ask an embryo whether it's okay with its genes of its future children being permanently altered, but consent is even difficult to provide to parents because it must be informed consent. There are many unknown risks involved with germline gene editing, so much so that it is difficult to communicate precise information with parents. In the case of He Jiankui, not much is known about how parents were consented, but the research was advertised as a way to have children with HIV prevention, as well as be a part of a scientific discovery. All of these ethical questions arise from germline gene editing for disease, but theoretically there are no limits to what CRISPR-Cas9 can be used for. This brings up another huge ethical issue of eugenics, or selectively breeding humans for traits deemed as desirable. Historically, eugenics has been used to wrongly suggest that some races are superior to others. In the context of CRISPR-Cas9, humans could change DNA to remove traits subjectively regarded as undesirable. Anything ranging from hair color to skin color, or even predisposition for mental or physical health could be targeted and changed with CRISPR-Cas9. But the scariest part is not imagining these possibilities. It's knowing that it's only a matter of time before someone tries it. Unless, of course, humans decide to enforce some ground rules. This is where the lovely politicians come into play. Different countries have varying regulations on CRISPR-Cas9, and many are running behind on the rapid progress this tool is making. However, there has been some international organization, such as through the 2018 Second International Summit on Human Genome Editing, in order to provide suggestions for governance. In the US, as we discussed previously, the FDA has approved CRISPR-based drugs, and there are similar drugs currently in clinical trials. However, there is still no federal legislation at all regarding CRISPR-Cas9. And sure, with an average Senate age of 64, we might have some ways to go on that front. Unfortunately, though, we might not have as much time as we think. Even while I'm writing this essay, the research continues, even outside just human gene editing. Between writing these words, I see that a company managed to create extinct dire wolves after 10,000 years using CRISPR technology. In another corner of the world, a biotechnology is working on lowering heritable high cholesterol in humans. And in China, we return to Dr. He Jiankui. While we might not know what he's up to now, the world will know soon enough. Should humans have this power? That remains to be seen. But it's up to us to be as well informed as possible on the capabilities of CRISPR-Cas9 so that we can quickly create human-centered policy. We should advocate for expert involvement in these policy decisions so that this technology can continue to be explored while also accounting for the serious ethical issues that arise from it. Our future selves will thank us for acting now. To conclude, I'll end with a quote from an unlikely source. The show Arcane deals with a lot of the same themes we discussed here regarding scientific advancement. When one of the main characters, Victor, becomes obsessed with the science that eventually consumes him and results in mass extinction in his world, he's left only with his thoughts in a desolate world. And in the decades he has to think about the consequences of his hasty ambition, he comes to a single, simple conclusion that I'll leave you with here. There is no prize to perfection, only an end to pursuit.